Greetings, class, and welcome back. When we last ended, we finished up looking at this slide here, which was what we talked about in the first lesson, which was glycolysis. We talked about the process of glycolysis, the beginnings of respiration. We talked about what it was, breaking down glucose and turning it into pyruvate. We talked about the input of ATP and then the net gain of 2 ATP as a result of this. We also talked about the production of NADH for the electron transport chain. Then we talked about the choice needing to be made, whether or not oxygen was present. If oxygen was present, aerobic respiration continues in the mitochondria. If oxygen is not present, then cellular respiration will become anaerobic and it will kick back into glycolysis again. But what we're going to look at today is if oxygen is present. And so we're going to move on into the mitochondria. This is the mitochondria. This is where the Krebs cycle and the electron transport chain occurs. The mitochondria is a double membrane structure, as you can see. It has fluid, this fluid-filled cavity here in blue, that's called the matrix. And then it has these what are called inner mitochondrial membranes with folds. Those are called the cristae. These two locations are very important for what we'll be talking about today. The mitochondria is analogous to the chloroplast in that it's an energy transformation organelle, and that it's going to be taking energy in one form and transforming it into another that can be used by the cell. Remember, the mitochondria does not make energy. It's taking energy that already exists and it's transforming it into a different form that the cells can use. So what we're going to look at today first is the Krebs cycle. The Krebs cycle was named after Hans Krebs. It's also more commonly known as the citric acid cycle because that's the name of the compound that really powers the reaction. It occurs in the mitochondrial membrane. And what happens during the Krebs cycle is the pyruvate molecules that were produced during glycolysis are converted into carbon dioxide, NADH, FADH, and also produces a little tiny bit of ATP, which we'll be looking at in a moment. Okay, but again, the Krebs cycle does not create a lot of ATP, just like glycolysis did it. Again, it's about doing this, creating these electron carriers, the NADH and the FADH, which are going to eventually move on to the electron transport chain, just like the NADH did during glycolysis for the ultimate part of cellular respiration in the ETC, where the serious amount of ATP are produced. So let's take a look at the Krebs cycle or the citric acid cycle. The Krebs cycle begins with the entry of the pyruvate molecule right here, as you can see, into the matrix of the mitochondria. Of course, oxygen, remember, has to be present for this part to occur. As you can see, the pyruvate or pyruvic acid is slowly broken down. Carbon molecule is broken off to produce a little bit of CO2. And at this time where the pyruvic acid is broken down, an NADH molecule is produced, and you can see that NADH molecule is going to travel to the electron transport chain. That 2-carbon molecule will then join up with this 4-carbon molecule that's swinging around as the citric acid cycle spins. With the benefit of an enzyme called, or coenzyme called acetyl coenzyme A. That's the remains of this 2-carbon molecule. That 2-carbon molecule is going to join up with the 4-carbon molecule that's swinging around and create a new compound called citric acid. That citric acid is the compound that starts the citric acid cycle, also known as the Krebs cycle. It's a 6-carbon molecule, and as it travels around, you'll see it actually slowly starts to break down. As it releases carbons, it creates some carbon dioxides, but it also creates something else very important. As those bonds are broken, it releases high-energy electrons, some hydrogens to produce NADH here, NADH here. Also, you can see towards the back end a little bit of FADH being created, and also some ATP being produced here during the reaction. And when the Krebs cycle finishes, as it's completed its spin, you can see it's now a 4-carbon molecule again, looking for that acetyl coenzyme A, with the 2-carbon molecule coming from the next pyruvic acid molecule as it moves in. So this spin here that you'll actually see in the diagram represents one spin of the Krebs cycle. And remember, per glucose, you're going to get 2 pyruvates. And so this is going to get 2 spins per glucose molecule. So these numbers here that you're seeing are really only half of what's produced during the Krebs cycle. And let's look at a better diagram for that. This is a better diagram here. You can see the actual numbers. This one represents an entire glucose molecule. So the Krebs cycle in total, by the time it's produced everything for the glucose molecule. Remember, 2 pyruvates are going to come in, so you're going to get 2 different spins here, 1 for each. You're going to end up with 6 carbon dioxide molecules, 2 ATP. Not a lot, but you're going to end up with all this stuff over here. 2 NADH coming from the beginning when the pyruvate is entering into the matrix, and then also 6 more NADH as a result of the spinning of the citric acid cycle, and then 2 FADH, which are produced during the citric acid cycle. All that stuff, of course, is going to move to the electron transport chain. These compounds here are so important for what's going to happen later. Remember, the Krebs cycle, glycolysis, they were really about setting the stage for the last part of photosynthesis, which is the electron transport chain. That's where the bulk of the ATP are going to be produced. So let's look at the electron transport chain. The electron transport chain takes place in the cristae along the folds of the inner mitochondrial membrane. That's where all the NADH and FADH are going to end up. Those NADH and FADH are going to be carrying some very important passengers. They're going to be carrying high energy electrons and also the hydrogens, which are going to be used later to power the production of ATP. This process is also known as oxidative phosphorylation, but we call it electron transport chain. This is what the process looks like. You can see NADH coming in, NADH coming in. You can see FADH coming in. These, of course, came from the Krebs cycle. There was something else produced during the Krebs cycle. Remember that carbon dioxide gas? That's the carbon dioxide gas that you exhale when you breathe. But when you breathe, you also inhale gas, and that gas you inhale is oxygen. And this is where the oxygen comes in place. So now you're starting to see how all the parts come together for the equation. We already took care of the glucose, the raw material, from when you ate. We've taken care of the carbon dioxide as a product. We've taken care of the oxygen. We're inhaling that. We're going to use that now. Okay, and now we're going to look at the last part, which is that ATP energy, all that high ATP energy that's going to be produced during this part of the process, and one other waste product that's going to be released, which is water molecules. So I'll explain all that. So what's going to happen in the ETC is very similar to what happened, if you remember, in the light-dependent reaction. You have these high-energy electrons moving along the membrane here. And along this membrane, you have all these protein pumps, just like you did in the light-dependent reaction. But we don't have the sun's energy this time. Instead, we have the NADH and the FADH bringing the high-energy electrons that are going to power these pumps, because what's going to happen is these hydrogens are going to try to move from an area of low concentration to high concentration, which, remember, requires energy. And so those electrons are going to provide that energy. And as they go along, of course, they're losing a little bit of energy, and they need to be getting rid of. They're gotten rid of through the oxygens. The oxygens are the receptor that are going to pick up those high energy electrons and pick up some hydrogens and get rid of them. And they're going to get rid of them in the form of water molecules. So this is where the water is produced. As the oxygen picks up those high energy electrons and hydrogens, it creates water molecules. And those are also exhaled when you breathe. Next time you exhale, hold up a mirror or a piece of glass and see if you don't see a little bit of water vapor. That's the water. Again, those oxygens are very important because they have to pick up those electrons that have been spent, pick up some of them hydrogens, and get rid of them. But the goal of the electron transport chain is similar to that of the light-dependent reaction. It's creating an imbalance of hydrogens on one side. So you have this imbalance of charges. You have a high concentration of high energy hydrogens on one side, and then an imbalance with the other side where there's only a few. So these hydrogens are going to want to move back, and they do. They go through a specific place. That place is called ATP synthase, which is a structure that rotates like a turbine, and it rotates as the hydrogens flow through back into the matrix. They create almost like a water flow, like it would, say, with a dam, a hydroelectric plant turning that turbine to generate electricity. But instead of generating electricity, these flowing hydrogens are turning this turbine, this ATP synthase, to produce ATP, and lots of it. Lots of ATP is produced during this part of cellular respiration. This is why all that NADH and FADH was so important, because it was bringing the electrons and the hydrogens to power this part of the reaction. So this is what it looks like overall. The whole process of aerobic cellular respiration, remember, began with glycolysis. Remember, glycolysis produces 2 pyruvate molecules right here. We'll throw them right there. Those pyruvate molecules, of course, as they move into the matrix, will release some carbon dioxide gas as they begin to enter into the Krebs cycle. You can see there's the carbon dioxide gas that's produced during the Krebs cycle. That's exhaled by you. The Krebs cycle, of course, produces a little tiny bit of ATP, just like glycolysis does. But remember, glycolysis is more about producing electron carriers, the NADH. And the Krebs cycle is also really about producing electron carriers. Remember, it produces NADH and FADH, which are both going to go towards the electron transport chain to power that last part of the reaction. Here's that oxygen. You just took a breath breathing in while you're listening to me. That oxygen, of course, is going to be the final receptor for those spent electrons and some of those hydrogens. This process will produce 6 water molecules, which will be waste vapor that you'll exhale. But then ultimately, of course, you'll get plenty of ATP, about 32. Remember, there's not really an exact number, 32 to 34, and anywhere between 36 to 38 to 40-ish in terms of your ATP in total, because you got 2 from glycolysis, 2 from the Krebs cycle, and 32 from your electron transport chain. So like I said, between 36, 38, there is no, like I said, exact number, depending on what textbook you read, who you listen to. It's very hard to ascertain an exact number. And then this is what the process looks like. Also, this one is from your textbook. This one is broken down nicely. You can see the glucose here, of course, the pyruvate here, that little bit of ATP produced. Of course, we know what's going here, NADH. We also have the Krebs cycle here producing 2 ATP, giving off a little bit of CO2. But also, remember, joining in here is some NADH and FADH to power the electron transport chain, which, remember, is going to produce a little bit of water vapor because these oxygen molecules are going to be picking up the electrons and some of the hydrogens to produce 32 more ATP through ATP synthase, giving us, like I said, a total of about 36 to 38 total ATP produced very, very quickly over and over again by your mitochondria in your cells.